Welcome to Shaping Science, a collection of videos from the voices that are shaping science today. I'm Laura Hemmingham from Technology Networks, and today we're joined by Dr. Tad George, Senior Vice President of Biology R&D at Raresight. In this episode, we'll explore how spatial proteomics, including multi-plex imaging technologies like Raresight's Orion platform, are transforming translational research. So, without any further ado, let's get into the science. Hi, Tad. Thanks for joining us today. It's great to meet you.
Hi, Laura. You as well.
So today we're talking about Orion, Raresight's high-plex, high-resolution imaging platform designed for spatial proteomics. So to begin with, how does Orion fit into today's multi-omics workflows? And what advice would you give researchers just starting out with spatial proteomics?
Yeah, sure. That's a big question, and it's pretty relevant now these day and age because there has been a lot of discovery work that's been done with sort of spatial transcriptomics, and there's actually been some difficulty actually translating that to the clinic. So if we back up a little bit and talk about spatial biology discovery activities, they're typically focused on identifying sort of candidate protein targets that impact, health status through their influence, on the density and functional activity of cells within microenvironments. This is kind of when you're trying to move spatial biology into the clinic. Discovery level data, is typically obtained from really information-rich data sets, either high-plex RNA and or high-plex protein studies, executed on pretty low volume studies in which the emphasis is really on maximizing information content per sample. And sometimes people are even doing multi-omics in this space, right? It's like the integration of spatial transcriptomic data with kind of more targeted proteomics data. And when you transition, typically you do leave RNA behind because RNA expression, reflects functional potential, right? But protein expression actually dictates, actual phenotype and function, which is more important when you're doing clinical studies. So, yeah, like the multi-omics approaches are often focused on validation of discovery level leads that are revealed by RNA assays with multiplex proteins for translational applications. So, discovery work is often upstream, to reveal clinically relevant protein biomarkers that need to be validated at the protein level. And in the clinic, you need to do that in a large cohort study. So when people, we will talk a little bit more about Orion, but we do have users that are kind of integrating that with Orion. It's more common for researchers to sort of validate the transcriptomic hits by IHC and then build a multiplex Orion panel to process large cohorts of data with, tied to clinical outcomes, to derive those clinically relevant spatial biomarkers. So, but kind of I guess, getting back to the main question, translating this discovery-oriented data into, protein biomarkers with actual prognostic or therapeutic value, it kind of involves transitioning, to more targeted protein panels, and studies where clinical relevance, data quality, per sample run costs, throughput are paramount. So if you're going stepwise into this, you first need to sort of decide which cell types and states you want to analyze, and developing a quantitative analysis strategy. And both of these things drive selections of a smaller set of biomarkers that are included in a panel. And a common method, people employ is they'll use bioinformatics on those high-plex data sets to determine kind of a minimal set of biomarkers that adequately reveal what's going on in the microenvironment, in order to separate the populations and states that are relevant to the pathology that's being studied, right? And that means you have to develop and validate reagents used in the panel with control tissues, which often first involves identifying clones for each biomarkers that provide the expected staining results in a gold standard method like IHC. And then you have to validate that biomarker expression as detected by immunofluorescence to make sure it matches the IHC pattern. Once that's done, you have to validate the multiplex panel to make sure that it adequately resolves the cell types and states in the microenvironment that you want. which is usually done with a small pilot study typically with existing samples, right? It's typically a retrospective, samples that have been archived that have known clinical status and outcome relevant to the trial that you expect to apply the panel to. And it also involves making sure your analysis strategy correlates with the expected results. And then the last step really is sort of applying that validated panel, to preclinical testing activities or clinical trials, which involves somehow coming up with, that microenvironment level quantification in really large, well-designed studies with spatial biology results tied to clinical outcomes, using throughputs and turnaround times that can get through that level. So that's a long answer, but I think that was kind of what came to mind.
Great. And then building on that foundation, how do researchers take spatial biology discoveries from the lab to the clinic?
Yeah, I think anytime you transition towards the clinical, you're kind of moving from generally, there's a lot more rigor and operationalization that's required, and if you think, and also just access to the right, samples and expertise. So if I were to, think of the big ones, first is, I would say access to high quality samples that are part of large, well-designed cohorts tied to clinical outcomes. A lot of people just don't have access to those samples or studies. And these types of studies include, typically prospective, preclinical testing and clinical trial studies, and drug trials, for example, as well as retrospectives. A lot of times there's tissue banks that have, known clinical outcomes, and you may want to, do some prognostic work. But those samples need to be available. They need to be high quality, and you need to be able to have a large number of them in order to get statistical rigor, because you're typically looking for Kaplan-Meier curve level, data. That's one thing. So another one is if you have access to the samples, you also need access to and successful deployment of the appropriate technology and expertise that can get through this data. So on the technology side, it's things like throughput, run cost, access to validated reagents, and a workflow that enables operationalization of large cohort studies, right? On the expertise side, typically you got to integrate pathologist, spatial, and medical knowledge with data scientists, computational and statistical knowledge, and finding a way to, make those two types of expertises flow together and interact with one another. So these are things you're not necessarily doing so much on the more discovery side. I mean, you should, but it's not required. I would say another one is, moving from an emphasis, I would say, on data quantity, which is more on the research side, to data quality. So you start stressing things like reagent and panel validation, dealing with lot-to-lot and run-to-run variation, putting in place a QC infrastructure to make sure your tissue quality is fine, the staining and scanning run performance went well, emphasis on accuracy and reproducibility, things that are, a lot of work that's not necessarily going to get you a paper, so to speak, but are really necessary to give you confidence that the results are reliable, right? You also, as I mentioned before, have to decide what biomarkers you're going to put in the panel, right? So, this is downstream of the more discovery-oriented data sets that are derived from higher plex studies, because it's not really, once you're doing super high plex, the cost and throughput goes way down. Is you need to decide what you're going to put in the panel. Sometimes you're moving from an RNA or nucleic acid-based world to protein detection. So, if that's new to you, it's actually, even though they're difficult analytes to detect generally, but development of reagents used for detecting like linear sequences, say DNA and RNA, with complementary sequences, is far more straightforward than what's required for antibody-based detection of tertiary epitopes or shapes that are perturbed by fixation and antigen retrieval, and sample processing protocols. So like the reagents, in addition, antibodies typically on a per analyte basis are much more expensive than, say, a sequence that you can synthesize with a sequencer, right? So therefore, multiplex protein detection, which is typically what you're trying to do in clinical, requires empirical determination of what ensemble of clones reveal the expected expression pattern with a common sample prep and antigen retrieval protocol. So, on top of that, I mentioned the price. So you have to be prepared to deal with that reality, right? And oftentimes the elephant in the room is developing an effective quantitative analysis strategy, so that you're not, even if you can operationalize high throughput staining and imaging, you certainly don't want to bottleneck the trial when you can't get those results, turned into a quantitative result that's meaningful. So I guess those would be the big things that I think are part of that obstacle course, the hurdles you have to run to if you're trying to go into that translational clinical space.
Right. And of course, translating discoveries comes with its own challenges. What are the biggest hurdles researchers face when bringing spatial biology into clinical or translational settings?
Yeah, I think that it does dovetail kind of with those hurdles. It's, basically advice is how do we get through those hurdles, right? So I think the cornerstones for success, in applying spatial proteomics in the translational or clinical setting is, you know, you want to have an emphasis on data quality. You need the right throughput. You need panel flexibility. You need favorable cost. You need workflow convenience. And a lot of these things kind of work together. So like throughput and cost work together, because the faster you can run at a reasonable cost, two things happen. The more users at the site will have access to the technology, which is really important for multi-core user core facility settings. So even if you're not involved in clinical trial stuff, maybe you are more on the research side, but if you have people that can afford to use the system, you'll get more and more data just even on small cohort studies. But of course, if you're applying a larger cohort studies, you need to be able to afford the reagents for the study because you have lots of samples, and you want to make sure that the throughput is commendable with the size of the study. Throughput's also impacted by things like plex level, ease of panel design. You only bottleneck where it takes you a year to develop your panel, right? Speed of sample processing, imaging, and analysis, and also by access to what I would call workflow convenience and parallelization. So when you get to the high throughput studies, you typically want kind of a pathology oriented workflow where the staining is done in parallel to the imaging and analysis, meaning I might have samples coming in from a prospective trial this week. I'm going to stain those, but I'm already going to be scanning stuff from last week. I don't want to wait for the scanning to finish before I start my staining and imaging, right? So you start to have this typical, more, I would say, clinical sample testing workflow that people are using where they're staining in one area while scanning's happening kind of in parallel, right? And getting back to the plex level, it's really important to optimize the plex level required for the application and don't overplex, right? Because if you have a bunch of irrelevant biomarkers, it's just going to bog you down in your throughput and your cost, right? Back to, I think I mentioned panel design. It's typically performed first by validating individual antibody biomarker performance, on the platform in comparison to IC. So ideally you have, access to reagents or predefined panels or things that are already, you can already build from to begin with. Otherwise you have to make all of your own reagents, which can slow you down. I think we alluded a little bit to the complexity of spatial biology quantitative analysis. It's always good if you're getting in the space to either know how to do or have access to good computational resources like, computers, software experts, right, that are in this area. Yeah, I think, and then finally, you can't compromise on data quality, right? Because all of your quantitative insights are built on the foundation of the underlying data quality. That's impacted by the level of sample integrity that you can maintain through the process. through the staining and imaging steps by the capabilities of the scanning system. Is it sensitive enough? Does it have, the proper image quality, dynamic range, et cetera? And people just always forget this. The level of reagent validation is the foundation. So, so no matter what platform you're looking at, just be prepared to have validated reagents and don't skip that step. So that would be my answer to that question.
And despite these challenges, the tools themselves are incredibly powerful. Why is multiplex immunofluorescence such a powerful tool for translational research?
Translational clinical applications kind of require that spatial resolution at the cellular level, in microenvironments. So in that case, you're typically going to gravitate towards imaging-based technologies that can resolve, more than one protein biomarker at a time. So immunofluorescence is a very common and powerful technique that falls into this space. I mean, it's basically what immunofluorescence means is detecting biomarkers with antibodies, right? That's the immuno part, that are either directly or indirectly conjugated with fluorophores, right? Which is the fluorescence part. And in tandem with imaging systems that are able to sensitively and specifically detect that fluorescence with subcellular resolution that gives you that spatial knowledge. And multiplexing is enabled because the fluorophores have different, absorption emission profiles, and the scanners are developed to resolve those. You can typically do on normal microscopes, 3 or 4 different markers in a single scanning round. So it starts to push you towards the ability to kind of do the things we're talking about, where you need like maybe, you know, 5 to 15 or 20 different biomarkers analyzed in spatial context. Multiplex immunofluorescence has that potential. So that's kind of why it's used, I think, in translational.
Hmm. And with such powerful tools, the ability to capture more information at once takes research to the next level. Orion can capture 20 channels in a single scan. Why is that such a game changer?
Yeah, so kind of following up on the multiplex immunofluorescence before, traditionally, fluorescence overlap, limits the number of biomarkers that you can simultaneously detect, by normal scanners to somewhere around 3 to 5, which is far below the typical range of like 10 to 20 markers that are really needed to adequately resolve the cell types and states and biomarkers in the tissue microenvironment. So it's kind of, you have this challenge, right? So what prior to Orion, this led most technology advances down the road of cyclic staining and imaging, where you would iteratively, stain for 2 or 3 markers, image them, remove that signal, stain for the next 3, and so forth, which is really quite good at developing sufficient plex to get to that, those spatial answers. But it completely is just a dead end for the throughput required for the translational clinical large cohort studies, right? So the Orion imaging approach, what we did there, we had to invent a device that would expand the number of markers that could be imaged in a single round up to 20. So that, and we also made it the staining also the same way. It's staining in sort of one step without any amplification. So that platform kind of single round staining with direct conjugates combined with a scanner that can actually resolve 20 channels of fluorescence in a single scan really unlocks the throughput required at the plex required for the clinical translational applications. So that was not easy to do. It's a combination of instrumentation, software, and reagent advancements, essentially that worked with the sensitivity, specificity, and data quality required for translational applications. On the reagent side, we had to screen multiple floors to find ones that were bright, photostable, and had spectral spacing that maximized sensitivity and specificity. And they were also chosen specifically for ease of labeling, right? To make sure that the labeling didn't perturb the antibody's performance, such that IHC could always be reproduced in IF, and also such that you wouldn't be fully dependent on our company for making the reagents. It's actually easy for you to make your own reagents. So this was also, when you're thinking about reagent infrastructures, you got to think about how people are going to actually have access to the validation and products. We use the spectrum of the floors that we screen to actually, you know, source a laser box that was optimized for those floors. So the system has, multiple high-powered laser excitation on board for sensitivity with proprietary turnable, narrowband emission filters that are centered on those Argo floor peaks. And then we had to come up with an automated extraction algorithm to remove the residual spectral overlap, which you're not going to get away from with 20 channels and around. So the game changer part is that 20 channel in a single round is the sweet spot for the plex you need, but gives you the throughput, and run cost advantages and simplicity and data quality that's kind of required for those clinical translational applications.
Right. And beyond its capabilities is also about usability. What sets Orion apart from other multiplex imaging platforms, both in performance and ease of use?
Why don't we start with panel design? So the sort of reagent ecosystem that is part of the Orion world involves a couple of building blocks. One is, Argo floor conjugates that are pre-titrated and validated for use in the Orion system. And we have, I think, over 500 products available spanning, nearly 200 biomarkers, in the human and mouse space that you can buy directly from us that we've already validated to work in multiplex using the protocol that works for the Orion system. And I can't overstate the importance of that because, there's lots of clones that work, but they all need to work together as an ensemble and as an entire portfolio. Like we can't have, or it's not so ideal if 10% of our conjugates work well at low pH, the other 20 work at high pH, and if you want to put these together in any kind of order, then you're on your own to change the antigen retrieval and optimize. So no, all of these reagents are pre-titrated to work on the Orion with the antigen retrieval condition that we use all the time. We also have off-the-shelf panels for both human and mouse as opening building blocks for building panels. And I would say even internally, we've developed probably over 250 custom panels, for our services group, as well as working with our customers. So it's very easy to develop panels, and those can be used as building blocks for your, new panels. We sell the dye kits, so you can label your own reagents for custom biomarkers that don't exist in the catalog. It's super easy to do that. We also provide services if you want us to do it. And then we have a panel design tool, which allows you to configure custom panels for any application. So that's kind of ease of use on developing the panels, which is one of those major hurdles to getting into translational, is doing that effectively. I would say in terms of the pathology-based workflow, the workflow has extreme convenience of staining, imaging, quantitative analysis that are all done in parallel. I mentioned that before, is that it's very common for us to stain this week's samples while we're scanning last week's samples. The other thing I didn't mention is the quantitative analysis is done on board, and it's done once a scan is made, it starts performing the quantitative analysis, segmentation, feature calculation, etc., while it's scanning the next slide. So you don't have this sort of thing, I got to move all my data to network attached storage and then do a high-powered computer to the quantitation. It's all done kind of on board and in parallel with everything. So you have this parallelization. We also, the system has an option for, continuous 24/7 operation with the 30-slide autoloader, which is the software is designed not only just to run, multiple samples unattended, but it allows you to add and take off slides throughout the day and throughout the week as samples come in and as samples are done scanning. So you can think of it as it's all the imaging system is just continually generating data while in parallel you're staining new data. All these things are important when you're thinking about ease of use for operationalization of high volume situations. We also keep the pathologist in the loop. for sure. So that in the analysis, not only is the segmentation, feature calculation, and classification come with the platform, and what I should mention here, you don't have to use our segmentation, feature calculation, classification. You can use your own pipelines, but we do provide it. But our software also has hooks for the pathologist to be in the loop where they perform ROI-based annotation to say, this is where we need to make the analysis and do the quantitative. And that's really important for integrating pathologists in the workflow because, again, when you're doing the translational clinical stuff, you need their expertise. It comes with a really nice data management system, which allows remote access both to scanning and analysis and imaging. It has lots of built-in QC reports that are useful for core facilities, CROs, et cetera. So that's kind of the, I would say, ease of use type of stuff.
Amazing stuff. Tad, thank you so much for joining us today. It was a pleasure talking with you.
Yeah, you as well. Thanks, Laura.
Thank you. To learn more about spatial proteomics, multiplex immunofluorescence, and the technologies enabling translational impact, check out the links in the description. Thank you for watching. Together, we are shaping science.